Uh, Thank you, Elizabeth. The meeting is being recorded, just so you remember. You have most of the interventions today will be in English. But as you know, we have the translation, so you have different canales, different in the translation button, you can go into listening to English or to Spanish, or not at all if you understand both. So with no further ado, we are doing this webinar in the context of Closer, the project we are a lot of us involved in. And today the subject is data management, quite important for patients in rare diseases, and also even more, I guess, after all this COVID crisis, data is on everybody's mouth. So next slide, please. Because I don't know if, okay, thank you. So anyway, Closer is a great project. A lot of you are from the project in the audience today, so you know who you are, but we are several partners, both from Europe and Latin America, working all together to improve the way childhood leukemia is treated. Next slide. Sorry, it's a little hot here in Barcelona today. So anyway, the linking Closer between our different partners are the research, the people, and this partnership try to really generate impact and improvement in terms of diagnosing, treatment, and therapeutics in the future for childhood leukemia. Next. So just a very brief image of what is Closer. Again, how it came together, a group of very nice and dedicated people in different fields, patient association, clinician, researchers, working together and looking for ways to improve the current situation around childhood leukemia, especially in terms of the lack of epidemiological data, the different needs in research across different countries and hospitals, the current existence of complex methodologies for diagnostic and how we can improve those and make them more available, and also overcoming lack of resources, and also the approach and connection with the research in this field. Again, trying to connect better patients, clinicians, researchers, and all people working in the field. Next. So just our speaker today, first of all, thanks to all the three of them for joining us. We have Anthony Moorman, that comes from Newcastle University, Elena, Tomás from ASSERTA, a company dedicated or really experienced in real world data evidence and data management, and also Dimitrios Athanasiou. He comes from World Duchenne organization. He's not directly involved in Closer, but we've been collaborating in the past, and I'm sure in the present and future, in ensuring that patients have a strong voice, especially related to data management. So I think with no further ado, I will give the floor to Anthony. I'm sure you can introduce your work and what you do and your vision in data management. So with no further ado, again, thank you everyone for joining, and Anthony, the floor is yours.
Thank you very much for the kind introduction, and also thank you very much to the organizers for inviting me to talk today about data management. It's not often that I get invited to talk specifically about data management, so this is a real pleasure because I've been doing this for the past 30 years, and so it's with some pleasure, as I said, that I can impart exactly how we've gone about this process in the research context over the last couple of years, over the last 30 years. Sorry, I've managed to make a mess of this slide. Apologies. Right, there's my first slide. Thank you very much. So, as has already been said, I work at Newcastle University, and I'm currently co-leading a group called the Leukemia Research Cytogenetics Group that was established 30 years ago, back in 1992, with the working hypothesis that integrating genetic biomarkers into the management of patients with acute lymphoblastic leukemia will improve outcome. So that's quite a grand hypothesis, and the infrastructure and data management setup that I'm going to describe really here is the brainchild of Lorna Secker-Walker, who is the lady shown on the right-hand side of this picture. And it was her idea that in order to be able to improve the outcome of patients with ALL, we needed to, on a very large scale, to incorporate genetic data into the basic patient management of patients with this disease. And so she decided that the obvious way of doing this was to establish a network whereby a central group within the UK could collect all the genetic data that existed on these patients, and then supplement that data with more sophisticated genetic data so that we could then analyze the data on a very large scale and use that data in the next clinical trial to improve patient risk stratification, i.e. identify those genetic abnormalities that were associated with a particular poor outcome and give those patients more intensive therapy, and then the opposite for those that were doing well. So in order to do that, she needed to set up a very large data infrastructure across the whole of the United Kingdom, which linked together the clinical trials units that were actually running the trials, and also all of the genetic laboratories that generated this standard of care data across the UK. So the first thing that she did was establish essentially how the data was going to flow around the country. And so this is illustrated on this slide here, and the group is shown down here by this red circle down here. And what she did is she linked in the clinical study group. So these are the clinicians who actually run the clinical trials within the UK. She managed to convince them that she should sit on their committees and therefore have access to clinical trial data. And importantly, patients were then consented to make sure that we were allowed to collect their data. And of course, once you're linked into this clinical trial study group, you can then link into the clinical trials unit themselves, and these are the people who are actually running the trials. So what happened is that once this had been established, we established a flow of data whereby every single time a patient was registered onto a clinical trial at the clinical trials unit, they would send us information about that patient, i.e. their name, their date of birth, and where they were diagnosed. We would then use that information to contact one of the 25 or so NHS diagnostic laboratories around the country and request from them all the standard of care genetic data that they had generated on that patient. They would then send us that genetic data. They would also send us potentially some genetic material, but I'm not going to talk about that latter part today. And we would register that genetic data on our central database. At the end of that trial, of course, we would then be able to feed that information back to both the clinical trials unit and also the clinical study group. So at the end of the trial, the clinical trials unit would not only have all the patient data, the treatment data, and the outcome data, but they would then have a robust, reviewed genetic information to incorporate into their analyses. So over a period of many years, we built up some very large cohorts of information. And so this enabled us to create a database of large sequential clinical trials with detailed patient, demographic, clinical, treatment, genetic, and outcome data. And we did this primarily on patients with children with acute lymphoblastic leukemia, but we've also modeled this system within pediatric AML, also adult ALL and adult AML, and to a much lesser extent, pediatric lymphoma. So we've replicated this model throughout several different diseases. So in order to make sure that we have robust data that we can feed into these systems, we had to establish a data management system. And this is a rather complicated slide, but the key thing is shown over here on the left-hand side. So every time we acquired patient data, this came from a clinical trials unit. So of course it was robust data in itself, so we weren't reinventing the wheel. All the data that we collected all the patient data was uploaded to a bespoke database that I'm going to describe on the next slide, which we called SIMS. And we would then use SIMS to track exactly what we were doing within the systems. We would use this database in order to dispatch request forms to the regional laboratories. They would then fill in these forms and return these forms to us. This was all done in paper originally, but electronically later on. We would then collect in these genetic reports and upload them onto our database. Again, the same database, and we would then check the data, verify the data, and code the data, and then store all the paperwork securely. So that was our kind of data flow as it came through the research group. We then established a whole series of systems in order to be able to cross-reference that data with other data sets around the country, including the leukemia, the Bloodwise Childhood Leukemia Cell Bank. So we used to provide genetic annotation to them. We would cross-reference our data against that held by the clinical trials units. We would make sure that we were always linking the right genetic data to the right patient. We had systems to resolve data discrepancies between what the laboratory was telling us and what the clinical trials unit were telling us, things like dates of birth, the name of the patient, the date of diagnosis. So this was all a well-worked routine to make sure that we were always matching the right genetic data to the right patient. And obviously this kind of system here requires a large personnel. So we were being very lucky enough to make sure that we had data managers, statisticians, and also cytogeneticists. But the key thing that we had throughout this whole thing was a bespoke database system. And this is just a couple of screenshots from that database system. And it just illustrates the need to be able to code and collect all this genetic data in a kind of logical way. So this database houses all our genetic and patient information. So we store the karyotype of the patient, we store each of the individual FISH results, we store the molecular results and the genomic results, each in different parts of the system. So we can then search the system in order to be able to undertake analyses. And over the years, these are some of our research highlights. So we've managed to collect a database that comprises about 30,000 patients from more than 15 clinical trials, again, with a large amount of data enabling us to look at the frequency of different genomic abnormalities, to look at genotype and phenotype correlations, and also prognostic studies. And we've also built up a biobank of material, which I know is not quite the topic of this talk, but really this has allowed us to perform a whole series of genetic and genomic abnormalities to improve the quality of this data. And using these data, we've identified numerous cytogenetic risk stratified. We've discovered new high-risk genomic abnormalities such as the IAM21, and we've been able to develop risk stratification algorithms and also to look at the clinical relevance of various copy number alterations. So all of this data has then been fed back into subsequent clinical trials. And because the data has been collected from clinical trials and has that kind of robust, secure knowledge, it's therefore easier to convince the clinicians to make treatment alterations in the subsequent clinical trial. And so the next slide here shows just an example of how we've managed to use this very large data set to develop a prognostic index, which can be used to deliver stratified medicine in pediatric acute lymphoblastic leukemia. So all this data has been published, but essentially what we did is we looked for a variety of different univariate risk factors, developed a multivariate model, identifying those that were most relevant to predicting relapse, derived a prognostic index, which can then be applied to individual patient data and assign patients an individual risk score, which is shown here, plotted over here. And this is actually data from 2,405 patients on our database. And you can see that each patient has a unique risk score using this equation, and it correlates very tightly with the risk of relapse, which is demonstrated by this red line here. And using this discovery cohort, we were able to define 4 unique risk groups, which have very distinct event-free survivals. And then we were able to validate this in an external cohort and show that using exactly the same prognostic index equation, you can derive risk groups that are of very similar size and have remarkably similar event-free survivals. So what we're now doing is looking to take this kind of model forward, and we've been involved in another EU data sharing project called Harmony, which is really kind of taking forward this idea of kind of very large scale data sharing and collecting robust clinical trial data. But on a kind of European scale. And this project actually goes beyond acute lymphoblastic leukemia and actually is collecting data from all hematological malignancies. But if you look at their workflow here, this is actually quite similar to the workflow that we established back in the UK in 1992. So we needed to make sure that we had robust sources. We had security in this particular project. They have gone for anonymization of data. You needed to have data quality evaluation, which of course is what we did by having that checking process, harmonizing the data to a common data model, or having a single database that holds all your data so that it's robust, and hence you get data that is ready for analysis. And we're using data within this platform on acute lymphoblastic leukemia in order to develop this European knowledge bank. And the idea behind this is really to pool data from multiple clinical trials across a very large time period so that we can then analyze to validate and explore risk factors, whether they're individual biomarkers or collective profiles across very large, robust data sets. And the aim of this particular project is to collect around 15 to 20,000 data sets. And this project is currently ongoing, and we've collected so far about 11 to 12,000, and we hope to hit our target within the next couple of months. So finally, what is our kind of involvement in CLOSER? So of course, the overall kind of objectives of the LRCG, or the Leukemia Research Cytogenetics Group in the UK, and the Harmony project is very similar to the CLOSER project. The CLOSER project, of course, is linking South America with Europe. So, of course, that's a novel dimension. But in terms of the data management, the data analysis, the ethics and the data sharing agreements, these are all common things that we've had to deal with either in the UK or within Harmony, and vice versa. So our role within Newcastle's role within the CLOSER project is really to provide access to large, well-annotated data sets, either at the aggregated level or individual patient data level, that can be used as a kind of comparator data set or a kind of baseline data set. And of course, the research objectives of us all is the clinical evaluation of risk factors, essentially in a wide spectrum of patient cohorts. So I think I've pretty much come to my time there. So I'll just end very quickly by thanking the very large number of people who've been involved in establishing the Leukemia Research Cytogenetics Group, particularly Lorna Secker-Walker, who I mentioned earlier, and also my colleague Christine Harrison. But the team involves a very large number of people, so it does take a huge amount of personnel to collect and verify this data over a very long period of time. And of course, it takes money. And we've been lucky enough to get a large amount of money over the past 30 years from a charity called Blood Cancer UK, which has funded the bulk of this work over that time period. So I'll end there, and thank you very much for your attention. I look forward to the next 2 talks and then a lively Q&A session at the end. Thank you very much.
Thank you very much, Anthony, for the presentation. I think it was very interesting to see your work and all this, how the data comes together. So next, I'm going to hand it over to Elena. I think you can get the control over the presentation. So the floor is yours again, about 15 minutes. Thank you, Elena.
Okay, thank you. Okay, thanks to ClickLab for organizing this webinar. I'm Elena Tomás. I'm the data science director at ASSERTA. I'm a hospital pharmacist and biostatistician. I don't have the control of slides. No. Yes, okay, right. It's working. In ASSERTA, we use clinical data to obtain knowledge that allows us to improve healthcare process. ASSERTA headquarters is based in Barcelona. We start in 2012. Since then, we've grown 300%. And we are a team of hospital pharmacists, computer and data scientists with years of experience in clinical practice and in data management. We are working in 20 different countries, and we manage 5 million clinical episodes. Okay. Our main activity is healthcare analytics. We use clinical, operational, and financial data to help with the decision makings of healthcare organizations. We help them to improve patient care delivery, health outcomes, efficient and safe use of resource and business operations. In particular, we advise healthcare organizations about drug use and lab test use. So we are used to managing sensitive data because we use data about health. So we have implemented data management strategies to deal with this kind of data, this sensitive data. Closer project mission is to improve the diagnosis and prognosis of children with leukemia. And to conduct this research, we need data. Data about patients, about diagnosis techniques, about treatments, and about health outcomes. We must understand that data is very valuable because we can get information and knowledge from data. And therefore, we must handle the data in a way that ensures its accuracy, security, and ethical use. For that, ASSERTA role within Closer project is to lead the data management process. Let's see what are data management goals. Is understand and support the information needs of the project, capture, storing the integrity of data, ensure the quality of data and privacy and confidentiality, prevent unauthorized or inappropriate access of data, and ensure that data can be used effectively to add value to the project. For that, we are using the data management framework published by DAMA International Organization. Data management has different stages. Let's speak about them. Start with metadata. We have to know the metadata of our data. Metadata describes the concepts that the data represents and describes to the relationships between the data and the concepts. We use second stage. We use reference and master data when we use, for example, international disease classification. We must control data security. It's a matter of defining security policies to know who can edit data, who can read it, or who can use it. In the next stage is data architecture. We must identify data workflows into the whole project. In data quality management, we define the data handling in such a way as to ensure data integrity and quality. And we need data modeling and design to discover, analyze, and scope project data requirements in the next stage. And later we represent all these requirements in a data model scheme. Oh, sorry. I go back. The next stage is data integration and interoperability. Is the ability for multiple systems to communicate with them. In the step of data storage and operation, we must define physical data storage and its management. With the next step, that is documents and content stage, we shouldn't forget documents and content that we don't identify as a data. For example, electronic documents like Word or Excel files. We must store, protect, and control access to these files too, like data. And finally, we must build and maintain the technical environment to perform data analysis and to extract knowledge from data. That's the data warehousing. Let's see some example of these stages. For example, about master and reference data. We define concepts and code that represent these concepts. For example, when we use disease abbreviation, one example, BALL represents B-cell acute lymphoblastic leukemia. Other example is the use of data dictionaries. In Closer project, people work from 8 different countries and from different institutions. So they are using different ways to store data. On the other hand, we must put together all that data to analyze it and obtain information and knowledge. Thanks to data dictionaries, we use the same words and codes to refer the same concept. In that way, we all speak the same language in our database, in Closer database. Oh, sorry. We designed the data architecture framework for Closer project. This framework is made of 3 main stages. First one, data acquisition. It's about how different institutions gather and store their data. Second one is data storage, Closer data storage. It's about how project institutions enter data in Closer central repository. And finally, data exploitation. That is about how data is used, who has access, and how they use this data. Closer management policies applies in all that in all these data architecture steps, as you can see at the bottom of this slide. Okay, and speaking about content, document and content. As I said before, we shouldn't forget documents content that we don't identify at first as data. For example, electronic documents like PDF files or Excel files. We must store, protect, and control access to these files too, like data. In Closer project, we are using SharePoint platform from Microsoft. What about legal, ethical, and cultural aspects? We've collaborated in writing guidelines about data anonymization in this project. We define data security policies that deals with data backups, access controls, etc. We describe techniques to ensure data quality. And we are working to control data use, ensuring that data are not used out of project objectives. And one example of that data management is, for example, the creation of surveys. We've created the surveys that other institutions in the project asked for. And the surveys use a secure hosting in Sant Joan de Déu hospital servers, because Sant Joan de Déu is the institution that leads the project, and we store the data in this institution. And monthly, we return survey data to the institution that asked for this survey, ready to be analyzed. In that way, we ensure that the quality of data is okay. We ensure the privacy and confidentiality, and we prevent unauthorized or inappropriate data access. That's almost all, but I would like to encourage people working with data to use data management guidelines. There are 2 or 3 more, the more important guidelines, but we are using guidelines published by Data Management International Group, and it's a very useful tool to help when you work with data. Thanks. That's all.
Thank you so much, Elena. It's even getting more in the technical aspects, but I think it's important because then we know how the data is really managed. So thank you for your presentation. And now I will pass on the floor to Dimitrios Athanasiou, as he's a patient expert, and especially in the data management we've discussed in the past. We collaborated in other projects really related to that. So Dimitrios, the floor is yours. I think, can you move yourself, or you can say next slide to Dani and she will move for us.
Dani will save me so I don't destroy the presentation.
Thank you so much.
Thank you so much. First of all, I'm very honored to be here and looking at this great proposal. My name is Dimitrios Athanasiou. Next slide, please, Dani. And I come from World Duchenne Organization. I'm here because I have a son with Duchenne. It's a neuromuscular terminal disease, rare. And you will be impressed how common things are in leukemia, in other rare diseases, and even neuromuscular. Next slide, please. These are my disclaimers and my disclosures. You can find my declaration of interest in the hyperlink there. Next slide, please. I have to give you a very high level of Duchenne as a disease, because although it looks very different, you might find a lot of similarities. So Duchenne muscular dystrophy is a disease basically that destroys your muscles. It starts in infancy. Everything in our body, almost anything in our body is a muscle. It's one of the longest genes, if not the longest, because of how many muscles we have. It has a lot of mutations, various types of mutations, phenotypes, genotypes, etc. And it has an involving natural history, because like pediatric cancer in general, we have a lot of off-label use that we don't record. And this affects the natural history of the disease, and that creates problems when we do clinical trials research or we try to provide care. Next slide, please. So what are the challenges that we face? And I'm sure you will find something that you can relate to. So we have many unknown in the patient management and care. We have a lot of discrepancies in regional care. So our LATAM region is working very close to the European region, the US region to catch up. We try to support also the disease trajectory that is not as standard as we think, the evolving standards of care, the clinical trial designs, a lot of data issues and silo attitudes, and this applies from patient organizations to research organizations, to hospitals, to clinicians, to researchers, and to other entities like industry. So these are common problems, pipeline issues, and a lot of failing products, exclusion of our patients that are on ambulant, and we have serious problems in access to innovative therapies, like most of you, and to labeling, but also to access to proper standard of care. Next slide, please. So we try to deal with these challenges, and we broke them down to some basic areas so we can focus. The one, it was, and the most foremost, it was the fragmentation of effort and the fragmentation of data. Diagnosis and care management, that we have differences in various regions, even in the same country, even the same city, in two different centers, you have differentiation. The basic research is lagging behind, off-label use extensive, problems with the drug development pipeline, regulatory issues, because of the data that we bring to the regulators, maybe they are not efficient to provide us approvals for our drugs, access problems, and policy, because everything at the end, all of this becomes to policy. Next slide, please. And history of the community, just to have an idea. The disease was first described, I think, something like 180 years ago. So it's impressive. In 1950s, we have our first organization founded. About 1980s, we have our small organizations around the world. In 2000, we have a good network. And I think in 2020, if you move to the next slide, Danny, you will see that we have something like 50 members in 40 countries. So it is a very vibrant and very, let's say, concentrated, effort-concentrated network. Next slide, please. What is the result of this effort? So in research, we invest 80 million euros or dollars, it depends, in research and care. We've increased our clinical trials from 60 to 70 in the last decade, which included also a lot of funding from our side, but a lot of data also. Patient input was incorporated almost in all clinical trials. We've been involved with our data and our patient voice in all the, let's say, discussions that had to do with our condition in EMA and FDA. We work very heavily to increase the health literacy of our people, and we train every year more than 100 patient experts around the globe, and especially with the topic on data ownership, data sharing, etc. Educational material has reached out to many languages in more than 10,000 families. Imagine that in Europe we have something like 25,000 families, and the same also to US, just to give you a perspective. And in care, we support and finance the international standards of care, which are for us is the Holy Bible, let's say, for our care, although it's not followed. And I think this is something we have experienced also. We have the certified centers that we know that our children receive the best care they can ultimately get, and we are expanding this program to a global level. And we have increased the life expectancy of our boys. Just to tell you that the mean age of our, because mostly our boys are getting the heavy type, let's say, of the disease, and we lose them. We lose them about at the age of 25 to 30, depending on the condition. But obviously, you understand if it's a muscle disease, that means that they cannot move anything. It's totally inhabilant. Next slide, please. So because of the work that we have done in the data and the use of data and the support and the financing, we managed to increase the life expectancy of our children by 7 years the last decade with no treatments. And from leukemia, I think you value what this means, maybe even more than we do. So we have two umbrella organizations. The one is the World Duchenne Organization. It does advocacy, policy, and regulatory work. We're part of the biggest networks. We're part of the pediatric committee, the EMA and the FDA. Also, we work with our colleagues in the US. Next slide. Where we're involved in various projects, and I picked some because they might be interesting. So Vision DMD, for example, is a drug development project. So we finance and develop our own drugs. Bind is about the brain function in the dystrophies, because we're also it's very important, the quality of life. Share for Rare, most of you might know it, is a project that it gives power to the patient groups to do their own research and power to the patients to own the data, because this is critical. Trials at home, remote clinical trials, because obviously we cannot expect that in every village, everywhere in the planet, we will have a center that our patients will go. A very interesting model about the EuroNMD Registry Hub, which is the European reference network. One of them is for neuromuscular. I will explain a little bit further, and various other projects. Next slide, please. So in 2015, we realized that the next battle is going to be on data. So we have built a patient-led foundation focused on data collection, data harmonization, and facilitation. That was the reason. The reason we built it, it was because we needed to increase the the access to the data. We had to break down the silos, but also we need to collect more data in order to use it for research and training and drug development. It has 3 pillars: data, research, and training. Next slide. A very interesting, maybe beyond as you can imagine, the community advisory boards, the data repositories that we use in the Duchenne Data Foundation, the patient academies, etc., is the Duchenne Data Platform that our Dutch organization have initiated, which is basically a patient locker that you can have your data and you can provide access as a patient to the person you want. But also this has the potential to have an artificial agent that will consult you what to do better in order to care for your child. Very innovative. It has been discussed also in the European Medicine Agency. So we can do things if we think outside the box. Because of the GDPR, especially in Europe, travel is difficult to move. The same happens between the countries. And since the owner of the data is the patient, we reverse the GDPR and we empower the patient to have full control of his data and where he wants to donate it, let's call it like that, or use it, and have results out of this. Next slide, please. Sorry if I'm talking too fast. My Mediterranean temperament maybe got the best of me. One item that it is interesting is this registry hub. So in Europe, we have 24 ERNs, reference networks for diseases. One of them is for neuromuscular diseases. The funding that we have received from the EU, it was limited. So we decided 3 patient organizations to match the funding of the EU in order to make a proper registry hub. What's important on this hub is that it collects and it merges different types of data. So patient-reported outcomes, patient-registered data. It is fair by design and it's open and accessible. And of course, the patient organizations and the governance of this. Next slide, please. And this is the result of our work. And I highlighted some of them just to have an idea where we need this data and why data management plans and understanding of our communities, how important is the knowledge of data. We use these results that we have from our work on disease prognostic models, in silico, artificial intelligence, HTA and the reimbursement discussions, care guidelines, regulatory guidelines, clinical trial simulations, platforms, patient-reported outcomes, everything you can imagine. At the end, although we finance from animal models to exoskeletons, at the end, everything is on data and you can negotiate on data. I'll try to speak slower for the translator. Sorry. Next slide, please. I don't have a lot of slides, just a couple of slides. So what we realized very early, and maybe I will, this is going to be my first question to our fantastic colleagues in CLOSA, is about FAIR. Most of you know FAIR. The ones that do not know FAIR, it comes with an acronym of the data should be findable, accessible, interoperable, and reusable. When it comes to pediatrics, when it comes to rare diseases, data has extreme value. The owner is the patient. Many people can use it, but nobody should silo it. So when we realized that, we actually have a dedicated department that does verification of data. So it makes sure that the data is findable, accessible, interchangeable, and reusable inside our organizations. And we put this declaration that is shared by many organizations, that we want all our data to be fair by design. Next slide, please. So where we are at the moment, we're still in a pathway that we have many successes and failures. We improve the quality of life of our children. They live longer, but we still lose them all by Duchenne. We try to balance the hype and the hope between our community, but in order to do so, we need trained community, trained people, and to understand our people the value of the data and ask something in return from that. Next slide. So what the future holds, and I don't think it holds only for Duchenne, but also it holds for leukemia and many other diseases, is that we need to create this data. We need to design our projects, and we need to create the data that can accelerate the science. We don't need any more silo attitudes. We need to be patient-centric, reliable, and interoperable, high quality. We need to foster our cooperation together with the other stakeholders, but on an equal level. We're not only the donors of the data, it's not altruistic. The first question that should come is what's there for me? And this is a question that as patient organizations, as researchers or as clinicians, we should be able to answer. What is there for this patient that will donate his data? What's he getting back? So we need to build a consensus on that, strengthen our advocacy, and obviously to improve access to our standards of care, but also pediatric medicines overall, especially for rare diseases, for our best part of our community. Next slide, please. And I think it's the last. It's a big thank you. And my board of directors, you can see them there from our global organization. Thank you so much, guys.
Thank you so much, Dimitrios. I think it's a really nice presentation to end up this data management webinar, because it gives us a good overview of what can be done also when the patients are really involved. And I think that's why we really wanted to have you today, because anyway, as you said, there are different diseases, but I think the struggles are very connected, and the finding the solution is also very connected. So I will again thank our three speakers. I think it was very interesting, and maybe we can take away the presentation. You can turn on your camera. You can ask the question in whatever language you want. So I would like to know, does someone has a burning question to start with the conversation? Okay, Dimitrios.
I have a burning question.
Yes.
Is the data fair inside the Closer project? So is it findable, accessible, interchangeable, and reusable by design? Because I know that REDCap can do it. We have done it in EuroNMD. So that's my first question. And what is the open access policy? Thank you. Because I saw these fantastic diagrams. I missed the patient word and I missed the open access. Thank you, guys.
So either Anthony or Elena or even Mireia, do you want to answer the question?
Elena?
Yes.
Hi.
Yeah.
Okay. Data management involve fair use data. When we speak about control access and use data only in project objectives, some of these objectives are to share this knowledge. Not all raw data, but the knowledge we obtain with this data it will to be shared. Mireia, if you complete.
Yes, I completely agree. I think that we stated in the grant agreement and in all the documents of Closer that we are following this, that our data will be fair, open, accessible, but as Elena says, is the results. Of course, there's a protection of the raw data, and there's a huge system to protect all the raw data, but all the results coming out of Closer will follow these fair data rules, let's say. So yes, it is something Closer is committed with.
Okay, so any question to our speakers?
I have several questions for each of them.
So just maybe
I'm sorry, I'm always asking.
No, you shouldn't be sorry. It's always good to ask questions. Just so you know, Dimitrios, because obviously Anthony and Elena, you know, Mireia Camos is the coordinator of the Closer project from San Juan de Dios.
And so a curious person, yeah.
Yes, and she put all of us, a lot of us together. So always very thankful for that opportunity that we've got to work together. So Mireia, please.
And I'm very thankful for this fantastic team. Anthony, just let's start with you. It's really impressive that you created such a practical and useful platform. And as you said, there's a lot of personnel and resources needed to maintain, to keep them. Could you, well, different questions about that, but how many people do you think it's needed as a minimal to keep a constant and a really practical infrastructure for a national, at the national level?
Well, that's a good question. So I think the kind of minimum number is probably 4, but it very much depends on how you set up your data flow. So we have as I mentioned, linked into the clinical trials units that run the clinical trials. So we don't spend any time collecting or verifying the clinical treatment and outcome data, because we just have an agreement with them that they will send it to us. So they do all that work, we get the benefit. There's no point reinventing that wheel, because it's just reinventing more work. So we have spent most of our time actually collecting the genetic data. So that's the bit that we have spent our time, and we have a data manager that manages that process. They're a full-time data manager. We have a geneticist and myself that check every single case that goes onto the database. And that's just the standard of care data. And then, of course, we have a statistician who actually does the analysis at the end of the day. And we have always had access to a computer programmer who will develop our database for us. And that has been sufficient to keep everything going. Where we have needed additional people on top of that is where we do additional genetic characterization centrally. And then the numbers of people you need goes up massively, depending on how ambitious you want to be. So obviously, our projects that have involved kind of relatively modest genetic annotation have taken, maybe, 2 or 3 people. But when we had very labor intensive projects doing, fish, we had, teams of 5 or 6 working on a full-time basis. So it very much depends, but the basic data collection and annotation, if you try to be clever about it and make sure you're not duplicating any work, then you can get away with about 4. And that's based on collecting data, probably about 600 new cases a year. But then, of course, once you have data, and as long as you're fair about it, as Dimitrios says, then the minute it's findable, people ask you for it, right? So that requires work as well. So we have always made our data very available to everyone in the UK, and through IBFM and other international study groups. So we spend a lot of time, sending out data to other projects, and we've, contributed individual patient data to dozens of projects over the years. Our database is not currently online, so you can't just look it up, but it's findable in the sense that it's we publish a lot, and the fact that we have this data is in the publications. We have an open access policy, and we've, as I said, we've contributed data. We've never said no to a project, basically. So that's how we've worked. So then the number of people, you need those data managers and statisticians to keep all those projects going. The more successful you are, the more you're asked to do. That's part of the problem.
And there would be a question for the 3 of you. This is about, it's very important, as you said, networking, because it's all based on a good network functioning from the clinical trial units, from physicians, and from parents and organizations, as I see the Dimitrios view, which is very interesting. So I guess that to create such an infrastructure, first, networking, choosing the right person that can provide and can, this is for Elena, harmonize all these data, because you're receiving from very different clinical trials. So maybe, as Elena said, this is a data dictionary, whatever, you may receive different type of data from different people speaking different languages.
Yes, and so so to come back to the first question, in terms of building a network, yes, this is, of course, is important, but what we've always tried to do is we've never tried to reinvent the wheel. So what we've tried to do is build on existing networks, because there are lots of networks out there. So there's no point trying to do something completely novel. So it's much better to link 2 existing networks together than try to replicate something extra. In terms of data dictionary, yes, You're absolutely right. The minute you have one clinical trial and then you move on to the next one, the data dictionary changes. Unfortunately, that's just life. And you can't, in my experience, change that. And if you try to design a single data dictionary that's going to, last forever, you're either never going to get off the ground, or you're just going to drive yourself crazy, because data changes, right? And that's just life. And that's why you need researchers to go back and double check. And what I do is I keep the data separately as it was originally given to me. So I have one data set for every single clinical trial. And then when we do a project, we merge it. And sometimes we merge it using changing data this way and that way, because, people's definition on what, complete remission is changes. I mean, the definition we're talking about now in IBFM is not the same as the definition that we were talking about when I, was much younger and had more hair, right? So it just changes. And if you don't try to embrace that change, then I think you get stuck, and you either you get yourself into a rabbit hole and you can't do anything, or you never do anything. And so what we've tried to do is store the data in its original format wherever possible, just accept it as it comes. And then when we want to do a research project or do a particular analysis, that's where we put the effort in to make sure that we interpret the original data in the most accurate way possible, always bearing in mind that no data set is ever going to be perfect, and you have to accept that. And no estimate of survival is ever going to be perfect. That's why you have 95% confidence intervals. So you just have to do the best you can, be logical wherever possible, be consistent, and explain it in your papers. And that's what we've always done. If we tried to invent the perfect data dictionary 30 years ago, I'd still be trying to do it. It doesn't exist, I'm afraid. So you've just got to be pragmatic.
May I ask Elena, do you want to complete also on that? Because I think Mireia also wanted to maybe, the follow-up on your question.
Yeah, thank you. Data dictionary is live, is have to change between project, but we need a minimum of same concepts because to analyze data, the data have to be homogeneous, and to measure, to analyze the same thing, we need a minimum of homogeneous data.
Yeah, but you probably need someone who translates what the principal concept of one clinical trial, and this is the same that they are put in a different name, but it is exactly the same concept. So you need a translator. I mean, I guess that you, Anthony, work sometimes as a translator of the
it's more
concept.
A translation dictionary.
Yeah,
maybe.
You are the dictionary.
Okay, may I, because we are just passing, maybe if it's okay for everyone, just a couple of minutes more. So I wanted to know if there are more questions. It can be asked in Spanish or in English, it doesn't matter, even if in the chat. If not, I had a question, and maybe to start with Dimitrios, just a quick insight, because we have seen, or at least I have seen the research project for the past, let's say, a change, a trend, and I think it's a very positive one. And I think it's very illustrated in Closer, because we have 3 patient association involved, and so how do you deal with that? How do you deal with the connection with the clinicians, the researchers, the companies? And I would ask the same to Anthony and Elena. Have you seen a change of the patient being more involved, both in aware of what they're giving, and also asking maybe more on receiving from the data? So maybe just a last kind of round before we close the webinar. So Dimitrios?
Maybe I should go last then, because this is an interesting question I would like to hear also. Yeah.
Okay. So Anthony, do you want to start?
Right. So in keeping with what I said before, we've never wanted to reinvent the wheel. So we get our patient involvement via what in the UK we call the NCRI Clinical Studies Group. So they're the group of clinicians that run clinical trials. So they have a patient representative on that committee. And I sit on that committee now. Christine and Lorna did before me. And so we have always presented our data collection and our research projects at that group, and have instantly therefore had kind of some level of patient involvement. So that's the first level we do. The second level that we have patient involvement is through our funders. So the Blood Cancer UK and also Cancer Research in UK, of course, have involvement from patients who sometimes collect money, sometimes want to know more. And so we organize lab tours for them frequently throughout the project. We've hosted more recently webinars to explain what we do to them, what we do with the data, etc. But now we're kind of moving into a more kind of interactive space. And this is where we are now wanting patients, either parents or patients, to actually be more actively involved in what we're doing and kind of designing the research. So we're approaching that in two ways. Firstly, via our kind of ongoing trial, which is the Altogether trial, which is being run across Europe. So we've recently put out a call to survivors and parents to be involved actively within the committee structure that we've established within the Altogether trial. So we have a science committee, we have a kind of protocol committee, we have a board, and so we're actively recruiting survivors and parents to those committees so that they can not just hear about what research we're doing associated with the trial, but actually be actively involved and tell us what they would like to see research being done. Obviously, we're never going to be fulfill all their requirements and their needs, but at least we will hear from them. And so we are hoping that that mechanism will bring forward parents who do want to be actively involved on a larger scale. So it has definitely evolved over the years. It is sometimes a struggle to make sure that we're contacting the right set of people. So there is no magic list of all the parents of the children on a clinical trial, or indeed a list of survivors. And that's our current sticking point. Exactly how do you get in contact with the people that may want to be involved? And maybe Dimitrios can give us some tips on how we can do that, sure, because that's certainly one thing that we'd be struggling with.
Okay. Elena, from your side?
Yeah, in ASSERTA, we work advising health organizations, and we don't have a lot of contact directly with patients, but we involve patients in our analysis. For example, when we need to use data about wearables or other devices that patients have to give their data to donor or to share its data. But basically we use financial, administrative, healthcare data, and don't have patient involved directly in our data, only with this example of wearables or other.
Okay, so it's good that we took you to closer, so you can have a real first experience, as we had in Chapter with patient association. Okay, so the closing one from Dimitrios before we close this very nice webinar.
Thank you. Well, I think I have a little different perspective. Maybe it's because of my community, let's say. So there are various levels. I think the discussion is not, at least for us, discussion is not how to get them involved, but the first, and interact with everybody. I think it's how you create a value proposition. So how I can persuade Anthony that he should use my patient experts for this development, for example. How I can persuade Elena that when you design a questionnaire that it's about outcomes or preferences, you should have somebody together with you design it and not having answering it. How I can persuade Mira that sharing of data is good for altruistic reasons. But in this project, I would love to know what is out there for me. You know, I'm a leukemia survivor. What's there for me? Not for my future, you know, generations or something. So this is something that we design very closely. And we do this also with regulatory work. So if patient organizations and patient experts, for example, regulate all the drug development in EMA, in the pediatric committees or the other committees that are approved in EU, maybe we are good enough to design a questionnaire. So I think that what we need is patient involvement in very early. I think we all will benefit from this. We'll save a lot of trouble, a lot of resources. For us, it's pretty simple. So we finance our research, we finance the drug development. We actually take percentage, if it is successful, back to our community so we can reinvest in that. And I think you can negotiate differently. Education is a key. What Anthony said is critical. So you need patient experts. I think you know also from this disease area, at least in our area, because we have discussion maybe, you know, sometimes, yes, we need a lay person or a naive patient. I can tell you that in our condition, most probably in leukemia, the naive parent is the one who loses his child. I don't think it's very different from leukemia. So we don't need naive patients. We need expert patients. And if they have the knowledge, and we have knowledge, or we develop the knowledge in data, then these are the people that we should work together with you for the benefit of all. So working together is the key. Education for both of us, how to involve the relevant people and how to develop the skills. But this is the future. This is where we're going. I think, as who said it? If you don't embrace change, Anthony, right? Change is stepping on you. So
Definitely. And I think, as you said, Dimitrios, I think because of, I know different communities of patients and you know, we've been collaborating together for quite some time now. And it's true that in some diseases area, it's, you know, the level of patient expertise is not the same, but it's growing. And I think at least in Closer, we have the chance to have 3 wonderful patient association and also building on their expertise and getting involved and more the co-creation, you know, and not just the getting the results. So I think I have to, I had a question. Okay, Miriam, last question, quick one, because we're...
Oh, okay, nice. Oh, yes, this is not a question, just to say thanks to these great speakers. And to comment that in Chile, we are far away from what you are saying about how to involve patients, but clearly we are learning from you. So thank you very much.
Thank you very much.
Anais, maybe I add something. Miriam, you will be surprised how far away we are also in Europe. But this is another discussion.
Yeah, no, definitely. It's there, but at least the path is set. And I think people are changing mindset, and especially in terms of data where we really can see the change. So I'm really happy to be part of this. So I will, because we're already 15 minutes above the schedule, but in any case, many thanks to the 3 of you for participating today. I think it was very nice to get those different views, and many thanks for everybody to attend the session. I think the next webinar will be anyway in September. So we have the summer break, I mean the European summer break, and we will recall. But in any case, I think again, thank you very much, Anthony, Elena, and Dimitrios for being here today and sharing on data.
Thank you.
That's it for us.
Thank you.
Thank you very much, everyone, for joining. Have a great afternoon.
Bye-bye.
Bye.
Thank you. Bye.
Gracias, hasta luego.